Meiosis is the process that sexually producing organisms use to create gametes. During meiosis, a diploid cell becomes 4 haploid cells. A diploid cell is a cell that contains 2 copies of the DNA, while a haploid cell is a cell that contains only 1 copy of the DNA. A cell spends most of its life in interphase. An interphase consists of 3 stages. These 3 stages are G1, S, and G2. G1 is the period where the cell is growing, and during this period, the cell performs other normal functions. S is the stage where DNA is being replicated through DNA replication to create another copy so that the cell has 2. During G2, the cell prepares for cell division. Cell division starts with prophase. The chromosomes form, and chromosomes are just supercoiled DNA, and they look like strands. Through the process of synapsis, these chromosomes form up into homologous chromosome pairs, and homologous chromosomes are chromosomes that are similar in size and shape, and they contain the same genes, although they may contain different alleles. For example, if the gene is eye color, one chromosome might contain the eye color blue, while the other contains the eye color brown. Another fancy terminology for homologous chromosomes is bivalent. You might hear that. So once these homologous chromosomes have paired up, crossing over occurs. And crossing over is when 2 chromosomes exchange alleles for the same gene. The point at which this occurs is called the chiasma. All the spindle fibers start growing towards the chromosomes, and the nuclear membrane starts to dissolve. During metaphase, the homologous chromosomes align in the center of the cell in a random orientation. This means that on one side you might have your mother's DNA, and on the other side your father's, and then for the next pair they're switched. So each cell gets a variety of DNA. The spindle fibers elongate and attach to the chromosomes. The next stage is anaphase, where the spindle fibers begin to shorten and pull the homologous chromosomes apart to opposite ends of the cell. During telophase, the spindle fibers disintegrate, and a new membrane forms around the chromosomes. The cell begins to split through a process called cytokinesis to create 2 separate cells. Next begins the second prophase. The 2 cells now both perform this, and the chromosomes move again towards the center of the cell. The spindle fibers reform, and the cell membranes break down again. The chromosomes again align in the middle of the cells, and the spindle fibers attach to them. The spindle fibers shorten, and these chromosomes are pulled apart into singular chromosomes. Spindle fibers break down, and the nuclear membranes reform around the chromosomes once more. The cells begin to split again through cytokinesis to form a total of 4 cells. At the end of this process, you end up with a total of 4 gametes, which are sex cells. It's the cells that you use to reproduce that contain each only half of the DNA of a normal cell.